Chapter 4 Cell Structure 4.1 Studying cells. By the end of this section, you will be able to do the following. Describe the role of cells in organisms. Compare and contrast light microscopy and electron microscopy. Summarize cell theory. A cell is the smallest unit of a living thing. Whether comprised of one cell, like bacteria, or many cells, like a figure 4.1, a. Nasal sinus cells, viewed with a light microscope, b. Onion cells, viewed with a light microscope, and c. Vibrio tasmaniensis bacterial cells, seen through a scanning electron microscope, are from very different organisms, yet all share certain basic cell structure characteristics. Credit A. Modification of work by Ed Uthman, M.D. Credit B. Modification of work by Umberto Salvanin. Credit C. Modification of work by Anthony D'Onofrio, William H. Fowle, Eric J. Stewart, and Kim Lewis of the Lewis Lab at Northeastern University. Scale bar data from Matt Russell. Introduction. Close your eyes and picture a brick wall. What is the wall's basic building block? It is a single brick. Like a brick wall, cells are the building blocks that make up your body. Your body has many kinds of cells, each specialized for a specific purpose. Just as we use a variety of materials to build a home, the human body is constructed from many cell types. For example, epithelial cells protect the body's surface and cover the organs and body cavities within. Bone cells help to support and protect the body. Immune system cells fight invading bacteria. Additionally, blood and blood cells carry nutrients and oxygen throughout the body while removing carbon dioxide. Each of these cell types plays a vital role during the body's growth, development, and day-to-day maintenance. In spite of their enormous variety, however, cells from all organisms, even ones as diverse as bacteria, onion, and human, share certain fundamental characteristics. Chapter Outline 4.1 Studying Cells 4.2 Prokaryotic Cells 4.3 Eukaryotic Cells 4.4 The Endomembrane System and Proteins 4.5 The Cytoskeleton 4.6 Connections between cells and cellular activities. Human. We call it an organism. Thus, cells are the basic building blocks of all organisms. Several cells of one kind that interconnect with each other and perform a shared function form tissues. These tissues combine to form an organ, your stomach, heart, or brain, and several organs comprise an organ system, such as the digestive system, circulatory system, or nervous system. Several systems that function together form an organism, like a human being. Here, we will examine the structure and function of cells. There are many types of cells, which scientists group into one of two broad categories: prokaryotic and eukaryotic. For example, we classify both animal and plant cells as eukaryotic cells, whereas we classify bacterial cells as prokaryotic. Before discussing the criteria for determining whether a cell is prokaryotic or eukaryotic, we will first examine how biologists study cells. Microscopy cells vary in size. With few exceptions, we cannot see individual cells with the naked eye. So scientists use microscopes, micro equals small, scope equals to look at, to study them. A microscope is an instrument that magnifies an object. We photograph most cells with a microscope, so we can call these images micrographs. The optics of a microscope's lenses change the image orientation that the user sees. A specimen that is right side up and facing right on the microscope slide will appear upside down and facing left when one views through a microscope, and vice versa. Similarly, if one moves the slide left while looking through the microscope, it will appear to move right, and if one moves it down, it will seem to move up. This occurs because microscopes use two sets of lenses to magnify the image. Because of the manner by which light travels through the lenses, this two-lens system produces an inverted image. Binocular, or dissecting microscopes, work in a similar manner, but include an additional magnification system that makes the final image appear to be upright. Light microscopes to give you a sense of cell size. A typical human red blood cell is about 8 millionths of a meter or 8 micrometers, abbreviated as 8 micrometers, in diameter. A pinhead is about 2/1000ths of a meter, 2 millimeters, in diameter. That means about 250 red blood cells could fit on a pinhead. Most student microscopes are light microscopes, figure 4.2a. Visible light passes and bends through the lens system to enable the user to see the specimen. Light microscopes are advantageous for viewing living organisms, but since individual cells are generally transparent, Their components are not distinguishable unless they are colored with special stains. Staining, however, usually kills the cells. Light microscopes that undergraduates commonly use in the laboratory magnify up to approximately 400 times. Two parameters that are important in microscopy are magnification and resolving power. Magnification is the process of enlarging an object in appearance. Resolving power is the microscope's ability to distinguish two adjacent structures as separate. The higher the resolution, the better the image's clarity and detail. When one uses oil immersion lenses to study small objects, magnification usually increases to 1000 times. In order to gain a better understanding of cellular structure and function, scientists typically use electron microscopes. 96 Chapter 4, Cell Structure Access for free at openstax.org. Figure 4.2. Most light microscopes in a college biology lab can magnify cells up to approximately 400 times and have a resolution of about 200 nanometers. B. Electron microscopes provide a much higher magnification, 100,000x, and have a resolution of 50 picometers. Credit A. Modification of work by GCG, Wikimedia Commons. Credit B. Modification of work by Evan Bench, Electron Microscopes. In contrast to light microscopes, electron microscopes, Figure 4.2b, use a beam of electrons instead of a beam of light. Not only does this allow for higher magnification and, thus, more detail, Figure 4.3, it also provides higher resolving power. The method to prepare the specimen for viewing with an electron microscope kills the specimen. Electrons have short wavelengths, shorter than photons, that move best in a vacuum, so we cannot view living cells with an electron microscope. In a scanning electron microscope, a beam of electrons moves back and forth across a cell's surface, creating details of cell surface characteristics. In a transmission electron microscope, the electron beam penetrates the cell and provides details of a cell's internal structures. As you might imagine, electron microscopes are significantly more bulky and expensive than light microscopes. Figure 4.3. These Salmonella bacteria appear as tiny purple dots when viewed with a light microscope. B. This scanning electron microscope micrograph shows Salmonella bacteria, in red, invading human cells, yellow. Even though subfigure, B, shows a different Salmonella specimen than subfigure, A, you can still observe the comparative increase in magnification and detail. Credit A. Modification of work by CDC, Armed Forces Institute of Pathology, Charles N. Farmer, Rocky Mountain Laboratories. Credit B. Modification of work by NIAID, NIH. Scale bar data from Matt Russell. Link to learning for another perspective on cell size. Try the How Big interactive at this site, http://openstax.org/l/cell_sizes. Cell theory. The microscopes we use today are far more complex than those that Dutch shopkeeper Antony van Leeuwenhoek used in the 1600s. Skilled in crafting lenses, van Leeuwenhoek observed the movements of single-celled organisms, which he collectively termed animalcules. In the 1665 publication Micrographia, experimental scientist Robert Hooke coined the term cell for the box-like structures he observed when viewing cork tissue through a lens. In the 1670s, van Leeuwenhoek discovered bacteria and protozoa. Later advances in lenses, microscope construction, and staining techniques enabled other scientists to see some components inside cells. 4.1. Studying cells. 97. By the late 1830s, botanist Matthias Schleiden and zoologist Theodor Schwann were studying tissues and proposed the unified cell theory, which states that one or more cells comprise all living things. The cell is the basic unit of life, and new cells arise from existing cells. Rudolf Virchow later made important contributions to this theory. Career connection. Cytotechnologist. Have you ever heard of a medical test called a pap smear? Figure 4.4. In this test, a doctor takes a small sample of cells from the patient's uterine cervix and sends it to a medical lab where a cytotechnologist stains the cells and examines them for any changes that could indicate cervical cancer or a microbial infection. Cytotechnologist, cyto equals cell, are professionals who study cells via microscopic examinations and other laboratory tests. They are trained to determine which cellular changes are within normal limits and which are abnormal. Their focus is not limited to cervical cells. They study cellular specimens that come from all organs. When they notice abnormalities, they consult a pathologist, a medical doctor who interprets and diagnoses changes that disease in body tissue and fluids cause. Cytotechnologists play a vital role in saving people's lives. When doctors discover abnormalities early, a patient's treatment can begin sooner, which usually increases the chances of a successful outcome. Figure 4.4. These uterine cervix cells, viewed through a light microscope, are from a pap smear. Normal cells are on the left. The cells on the right are infected with human papillomavirus, HPV. Notice that the infected cells are larger. Also, 2 of these cells each have 2 nuclei instead of 1, the normal number. Credit. Modification of work by Ed Uthman, MD. Scale bar data from Matt Russell. 4.2 Prokaryotic cells. By the end of this section, you will be able to do the following. Name examples of prokaryotic and eukaryotic organisms. Compare and contrast prokaryotic and eukaryotic cells. Describe the relative sizes of different cells. Explain why cells must be small. Cells fall into one of 2 broad categories, prokaryotic and eukaryotic. We classify only the predominantly single-celled organisms bacteria and archaea as prokaryotes, pro equals, before, kary equals, nucleus. Animal cells, plants, fungi, and protists are all eukaryotes, EU equals, true. Components of prokaryotic cells. All cells share 4 common components. 1. A plasma membrane, an outer covering that separates the cell's interior from its surrounding environment. 2. Cytoplasm, consisting of a jelly-like cytosol within the cell in which there are other cellular components. 3. DNA, the cell's genetic material. And 4. Ribosomes, which synthesize proteins. However, prokaryotes differ from eukaryotic cells in several ways. A prokaryote is a simple, mostly single-celled, unicellular, organism that lacks a nucleus, or any other membrane-bound. 98 Chapter 4. Cell structure. Access for free at openstax.org. Organelle. We will shortly come to see that this is significantly different in eukaryotes. Prokaryotic DNA is in the cell's central part, the nucleoid, figure 4.5. Figure 4.5. This figure shows the generalized structure of a prokaryotic cell. All prokaryotes have chromosomal DNA localized in a nucleoid, ribosomes, a cell membrane, and a cell wall. The other structures shown are present in some, but not all, bacteria. Most prokaryotes have a peptidoglycan cell wall and many have a polysaccharide capsule, figure 4.5. The cell wall acts as an extra layer of protection, helps the cell maintain its shape, and prevents dehydration. The capsule enables the cell to attach to surfaces in its environment. Some prokaryotes have flagella, pili, or fimbriae. Flagella are used for locomotion. Pili exchange genetic material during conjugation, the process by which one bacterium transfers genetic material to another through direct contact. Bacteria use fimbriae to attach to a host cell. Career connection. Microbiologist. The most effective action anyone can take to prevent the spread of contagious illnesses is to wash his or her hands. Why? Because microbes, organisms so tiny that they can only be seen with microscopes, are ubiquitous. They live on doorknobs, money, your hands, and many other surfaces. If someone sneezes into his hand and touches a doorknob, and afterwards you touch that same doorknob, the microbes from the sneezer's mucus are now on your hands. If you touch your hands to your mouth, nose, or eyes, those microbes can enter your body and could make you sick. However, not all microbes, also called microorganisms, cause disease. Most are actually beneficial. You have microbes in your gut that make vitamin K. Other microorganisms are used to ferment beer and wine. Microbiologists are scientists who study microbes. Microbiologists can pursue a number of careers. Not only do they work in the food industry, they are also employed in the veterinary and medical fields. They can work in the pharmaceutical sector, serving key roles in research and development by identifying new antibiotic sources that can treat bacterial infections. Environmental microbiologists may look for new ways to use specially selected or genetically engineered microbes to remove pollutants from soil or groundwater, as well as hazardous elements from contaminated sites. We call using these microbes bioremediation technologies. Microbiologists can also work in the bioinformatics field, providing specialized knowledge and insight for designing, developing, and specificity of computer models of, for example, bacterial epidemics. Cell size at 0.1 to 5.0 micrometers in diameter. Prokaryotic cells are significantly smaller than eukaryotic cells, which have diameters ranging from 10 to 100 micrometers, figure 4.6. The prokaryote's small size allows ions and organic molecules that enter them to quickly diffuse to other parts of the cell. Similarly, any wastes produced within a prokaryotic cell can quickly diffuse. This is not the case in eukaryotic cells, which have developed different structural adaptations to enhance intracellular transport. 4.2. Prokaryotic cells 99 figure 4.6. This figure shows relative sizes of microbes on a logarithmic scale. Recall that each unit of increase in a logarithmic scale represents a 10-fold increase in the quantity measured. Small size, in general, is necessary for all cells, whether prokaryotic or eukaryotic. Let's examine why that is so. First, we'll consider the area and volume of a typical cell. Not all cells are spherical in shape, but most tend to approximate a sphere. You may remember from your high school geometry course that the formula for the surface area of a sphere is 4 pi r2, while the formula for its volume is 4 pi r3/3. Thus, as the radius of a cell increases, its surface area increases as the square of its radius, but its volume increases as the cube of its radius, much more rapidly. Therefore, as a cell increases in size, its surface area to volume ratio decreases. This same principle would apply if the cell had a cube shape, figure 4.7. If the cell grows too large, the plasma membrane will not have sufficient surface area to support the rate of diffusion required for the increased volume. In other words, as a cell grows, it becomes less efficient. One way to become more efficient is to divide. Other ways are to increase surface area by foldings of the cell membrane, become flat or thin and elongated, or develop organelles that perform specific tasks. These adaptations lead to developing more sophisticated cells, which we call eukaryotic cells. Visual connection figure 4.7. Notice that as a cell increases in size, its surface area to volume ratio decreases. When there is insufficient surface area to support a cell's increasing volume, a cell will either divide or die. The cell on the left has a volume of 1 cubic millimeter and a surface area of 6 square millimeters, with a surface area to volume ratio of 6:1, whereas, the cell on the right has a volume of 8 cubic millimeters and a surface area of 24 square millimeters, with a surface area to volume ratio of 3:1. Prokaryotic cells are much smaller than eukaryotic cells. What advantages might small cell size confer on a cell? What advantages might large cell size have? 4.3 Eukaryotic cells. By the end of this section, you will be able to do the following. Describe the structure of eukaryotic cells, compare animal cells with plant cells, state the role of the plasma membrane, summarize the functions of the major cell organelles. Have you ever heard the phrase, form follows function? It's a philosophy that many industries follow. In architecture, this means that buildings should be constructed to support the activities that will be carried out inside them. For example, a skyscraper should include several elevator banks. A hospital should have its emergency room easily accessible. Our natural world also utilizes the principle of form following function, especially in cell biology, and this will become clear as we explore eukaryotic cells, figure 4.8. Unlike prokaryotic cells, eukaryotic cells have 1. A membrane-bound nucleus. 2. Numerous membrane-bound organelles such as the endoplasmic reticulum, Golgi Apparatus, chloroplasts, mitochondria, and others. And 3. Several, rod-shaped chromosomes. Because a membrane surrounds eukaryotic cells' nucleus, it has a true nucleus. The word organelle means little organ, and, as we already mentioned, organelles have specialized cellular functions, just as your body's organs have specialized functions. At this point, it should be clear to you that eukaryotic cells have a more complex structure than prokaryotic cells. Organelles allow different functions to be compartmentalized in different areas of the cell. Before turning to organelles, let's first examine two important components of the cell: the plasma membrane and the cytoplasm. Visual connection 4.3, Eukaryotic cells 101 figure 4.8. These figures show the major organelles and other cell components of a typical animal cell and b. a typical eukaryotic plant cell. The plant cell has a cell wall, chloroplasts, plastids, and a central vacuole, structures not in animal cells. Most cells do not have lysosomes or centrosomes. If the nucleolus were not able to carry out its function, what other cellular organelles would be affected? The plasma membrane. Like prokaryotes, eukaryotic cells have a plasma membrane, figure 4.9, a phospholipid bilayer with embedded proteins that separates the internal contents of the cell from its surrounding environment. A phospholipid is a lipid molecule with two fatty acid chains and a phosphate-containing group. The plasma membrane controls the passage of organic molecules, ions, water, and oxygen into and out of the cell. Wastes, such as carbon dioxide and ammonia, also leave the cell by passing through the plasma membrane. Figure 4.9. The eukaryotic plasma membrane is a phospholipid bilayer with proteins and cholesterol embedded in it. The plasma membranes of cells that specialize in absorption fold into finger-like projections that we call microvilli. Singular equals 102. Chapter 4, Cell Structure. Access for free at openstax.org. Microvillis. Figure 4.10. Such cells typically line the small intestine, the organ that absorbs nutrients from digested food. This is an excellent example of form following function. People with celiac disease have an immune response to gluten, which is a protein in wheat, barley, and rye. The immune response damages microvilli, and thus, afflicted individuals cannot absorb nutrients. This leads to malnutrition, cramping, and diarrhea. Patients suffering from celiac disease must follow a gluten-free diet. Figure 4.10. Microvilli, as they appear on cells lining the small intestine, increase the surface area available for absorption. These microvilli are only on the area of the plasma membrane that faces the cavity from which substances will be absorbed. Credit, micrograph. Modification of work by Louisa Howard. The cytoplasm. The cytoplasm is the cell's entire region between the plasma membrane and the nuclear envelope, a structure we will discuss shortly. It is comprised of organelles suspended in the gel like cytosol, the cytoskeleton, and various chemicals, figure 4.8. Even though the cytoplasm consists of 70 to 80% water, it has a semi-solid consistency, which comes from the proteins within it. However, proteins are not the only organic molecules in the cytoplasm. Glucose and other simple sugars, polysaccharides, amino acids, nucleic acids, fatty acids, and derivatives of glycerol are also there. Ions of sodium, potassium, calcium, and many other elements also dissolve in the cytoplasm. Many metabolic reactions, including protein synthesis, take place in the cytoplasm. The nucleus. Typically, the nucleus is the most prominent organelle in a cell, figure 4.8. The nucleus, plural equals nuclei, houses the cell's DNA and directs the synthesis of ribosomes and proteins. Let's look at it in more detail, figure 4.11. Figure 4.11. The nucleus stores chromatin, DNA plus proteins, in a gel-like substance called the nucleoplasm. The nucleolus is a condensed chromatin region where ribosome synthesis occurs. We call the nucleus boundary the nuclear envelope. It consists of two phospholipid bilayers, an outer and an inner membrane. The nuclear membrane is continuous with the endoplasmic reticulum. Nuclear pores allow substances to enter and exit the nucleus. 4.3. Eukaryotic cells. 103. The nuclear envelope. The nuclear envelope is a double membrane structure that constitutes the nucleus' outermost portion, figure 4.11. Both the nuclear envelope's inner and outer membranes are phospholipid bilayers. The nuclear envelope is punctuated with pores that control the passage of ions, molecules, and RNA between the nucleoplasm and cytoplasm. The nucleoplasm is the semi-solid fluid inside the nucleus, where we find the chromatin and the nucleolus. Chromatin and chromosomes. To understand chromatin, it is helpful to first explore chromosomes, structures within the nucleus that are made up of DNA, the hereditary material. You may remember that in prokaryotes, DNA is organized into a single circular chromosome. In eukaryotes, chromosomes are linear structures. Every eukaryotic species has a specific number of chromosomes in the nucleus of each cell. For example, in humans, the chromosome number is 46, while in fruit flies, it is 8. Chromosomes are only visible and distinguishable from one another when the cell is getting ready to divide. When the cell is in the growth and maintenance phases of its life cycle, proteins attach to chromosomes, and they resemble an unwound, jumbled bunch of threads. We call these unwound protein chromosome complexes chromatin, Figure 4.12. Chromatin describes the material that makes up the chromosomes both when condensed and decondensed. Figure 4.12. This image shows various levels of chromatin's organization, DNA and protein. B. This image shows paired chromosomes. Credit B. Modification of work by NIH. Scale bar data from Matt Russell. The nucleolus. We already know that the nucleus directs the synthesis of ribosomes, but how does it do this? Some chromosomes have sections of DNA that encode ribosomal RNA. A darkly staining area within the nucleus called the nucleolus, plural equals nucleoli, aggregates the ribosomal RNA with associated proteins to assemble the ribosomal subunits that are then transported out through the pores in the nuclear envelope to the cytoplasm. Ribosomes are the cellular structures responsible for protein synthesis. When we view them through an electron microscope, ribosomes appear either as clusters, polyribosomes, or single, tiny dots that float freely in the cytoplasm. They may be attached to the plasma membrane's cytoplasmic side or the endoplasmic reticulum's cytoplasmic side and the nuclear envelope's outer membrane. Figure 4.8. Electron microscopy shows us that ribosomes, which are large protein and RNA complexes, consist of two subunits, large and small. Figure 4.13. Ribosomes receive their orders for protein synthesis from the nucleus where the DNA transcribes into messenger RNA, mRNA. The mRNA travels to the ribosomes, which translate the code provided by the sequence of the nitrogenous bases in the mRNA into a specific order of amino acids in a protein. Amino acids are the building blocks of proteins. 104 Chapter 4, Cell Structure Access for free at openstax.org. Figure 4.13. A large subunit, top, and a small subunit, bottom, comprise ribosomes. During protein synthesis, ribosomes assemble amino acids into proteins. Because protein synthesis is an essential function of all cells, including enzymes, hormones, antibodies, pigments, structural components, and surface receptors, there are ribosomes in practically every cell. Ribosomes are particularly abundant in cells that synthesize large amounts of protein. For example, the pancreas is responsible for creating several digestive enzymes, and the cells that produce these enzymes contain many ribosomes. Thus, we see another example of form-following function. Mitochondria. Scientists often call mitochondria, singular equals mitochondrion, powerhouses, or energy factories, of both plant and animal cells because they are responsible for making adenosine triphosphate, ATP, the cell's main energy-carrying molecule. ATP represents the cell's short-term stored energy. Cellular respiration is the process of making ATP using the chemical energy in glucose and other nutrients. In mitochondria, this process uses oxygen and produces carbon dioxide as a waste product. In fact, the carbon dioxide that you exhale with every breath comes from the cellular reactions that produce carbon dioxide as a byproduct. In keeping with our theme of form-following function, it is important to point out that muscle cells have a very high concentration of mitochondria that produce ATP. Your muscle cells need considerable energy to keep your body moving. When your cells don't get enough oxygen, they do not make much ATP. Instead, producing lactic acid accompanies the small amount of ATP they make in the absence of oxygen. Mitochondria are oval-shaped, double-membrane organelles, figure 4.14, that have their own ribosomes and DNA. Each membrane is a phospholipid bilayer embedded with proteins. The inner layer has folds called cristae. We call the area surrounded by the folds the mitochondrial matrix. The cristae and the matrix have different roles in cellular respiration. Figure 4.14. This electron micrograph shows a mitochondrion through an electron microscope. This organelle has an outer membrane and an inner membrane. The inner membrane contains folds, called cristae, which increase its surface area. We call the space between the two membranes the intermembrane space, and the space inside the inner membrane the mitochondrial matrix. ATP synthesis takes place on 4.3, eukaryotic cells 105 the inner membrane. Credit. Modification of work by Matthew Britton. Scale bar data from Matt Russell. Peroxisomes. Peroxisomes are small, round organelles enclosed by single membranes. They carry out oxidation reactions that break down fatty acids and amino acids. They also detoxify many poisons that may enter the body. Many of these oxidation reactions release hydrogen peroxide, H2O2, which would be damaging to cells. However, when these reactions are confined to peroxisomes, enzymes safely break down the H2O2 into oxygen and water. For example, peroxisomes in liver cells detoxify alcohol. Glyoxysomes, which are specialized peroxisomes in plants, are responsible for converting stored fats into sugars. Plant cells contain many different types of peroxisomes that play a role in metabolism, pathogen defense, and stress response, to mention a few. Vesicles and vacuoles. Vesicles and vacuoles are membrane-bound sacs that function in storage and transport. Other than the fact that vacuoles are somewhat larger than vesicles, there is a very subtle distinction between them. Vesicle membranes confuse with either the plasma membrane or other membrane systems within the cell. Additionally, some agents such as enzymes within plant vacuoles break down macromolecules. The vacuole's membrane does not fuse with the membranes of other cellular components. Animal cells versus plant cells. At this point, you know that each eukaryotic cell has a plasma membrane, cytoplasm, a nucleus, ribosomes, mitochondria, peroxisomes, and in some, vacuoles. But there are some striking differences between animal and plant cells. While both animal and plant cells have microtubule organizing centers, MTOCs, animal cells also have centrioles associated with the MTOC, a complex we call the centrosome. Animal cells each have a centrosome and lysosomes, whereas most plant cells do not. Plant cells have a cell wall, chloroplasts and other specialized plastids, and a large central vacuole, whereas animal cells do not. The centrosome is a microtubule organizing center found near the nuclei of animal cells. It contains a pair of centrioles, two structures that lie perpendicular to each other, figure 4.15. Each centriole is a cylinder of 9 triplets of microtubules. Figure 4.15. The centrosome consists of two centrioles that lie at right angles to each other. Each centriole is a cylinder comprised of 9 triplets of microtubules. Nontubulin proteins, indicated by the green lines, hold the microtubule triplets together. The centrosome, the organelle where all microtubules originate, replicates itself before a cell divides, and the centrioles appear to have some role in pulling the duplicated chromosomes to opposite ends of the dividing cell. However, the centriole's exact function in cell division isn't clear, because cells that have had the centrosome removed can still divide, and plant cells, which lack centrosomes, are capable of cell division. Lysosomes. Animal cells have another set of organelles that most plant cells do not, lysosomes. The lysosomes are the cell's garbage disposal. In plant cells, the digestive processes take place in vacuoles. Enzymes within the lysosomes aid in breaking down proteins, polysaccharides, lipids, nucleic acids, and even worn-out organelles. These enzymes are active at a much lower pH than the cytoplasms. Therefore, the pH within lysosomes is more acidic than the cytoplasm's pH. Many reactions that take place in the cytoplasm could not occur at a low pH. So again, the advantage of compartmentalizing the eukaryotic cell into organelles. 106. Chapter 4, Cell Structure. Access for free at openstax.org. Is apparent. The cell wall. If you examine figure 4.8, the plant cell diagram, you will see a structure external to the plasma membrane. This is the cell wall, a rigid covering that protects the cell, provides structural support, and gives shape to the cell. Fungal and some protistine cells also have cell walls. While the prokaryotic cell wall's chief component is peptidoglycan, the major organic molecule in the plant, and some protists, cell wall is cellulose, figure 4.16, a polysaccharide comprised of glucose units. Have you ever noticed that when you bite into a raw vegetable, like celery, it crunches? That's because you are tearing the celery cell's rigid cell walls with your teeth. Figure 4.16. Cellulose is a long chain of beta-glucose molecules connected by a 1-to-4 linkage. The dashed lines at each end of the figure indicate a series of many more glucose units. The size of the page makes it impossible to portray an entire cellulose molecule. Chloroplasts. Like the mitochondria, chloroplasts have their own DNA and ribosomes, but chloroplasts have an entirely different function. Chloroplasts are plant cell organelles that carry out photosynthesis. Photosynthesis is the series of reactions that use carbon dioxide, water, and light energy to make glucose and oxygen. This is a major difference between plants and animals. Plants, autotrophs, are able to make their own food, like sugars used in cellular respiration to provide ATP energy generated in the plant mitochondria. Animals, heterotrophs, must ingest their food. Like mitochondria, chloroplasts have outer and inner membranes, but within the space enclosed by a chloroplast's inner membrane is a set of interconnected and stacked fluid-filled membrane sacs we call thylakoids, figure 4.17. Each thylakoid stack is a granum, plural equals grana. We call the fluid enclosed by the inner membrane that surrounds the grana the stroma. Figure 4.17. The chloroplast has an outer membrane, an inner membrane, and membrane structures, thylakoids that are stacked into grana. We call the space inside the thylakoid membranes the thylakoid space. The light harvesting reactions take place in the thylakoid membranes, and sugar synthesis takes place in the fluid inside the inner membrane, which we call the stroma. Chloroplasts also have their own genome, which is contained on a single circular chromosome. The chloroplasts contain a green pigment, chlorophyll, which captures the light energy that drives the reactions of photosynthesis. Like plant cells, photosynthetic protists also have chloroplasts. Some bacteria perform photosynthesis, but their chlorophyll is not relegated to an organelle. 4.3. Eukaryotic cells. 107. Evolution connection. Endosymbiosis. We have mentioned that both mitochondria and chloroplasts contain DNA and ribosomes. Have you wondered why? Strong evidence points to endosymbiosis as the explanation. Symbiosis is a relationship in which organisms from two separate species depend on each other for their survival. Endosymbiosis, endo equals within, is a mutually beneficial relationship in which one organism lives inside the other. Endosymbiotic relationships abound in nature. We have already mentioned that microbes that produce vitamin K live inside the human gut. This relationship is beneficial for us because we are unable to synthesize vitamin K. It is also beneficial for the microbes because they are protected from other organisms and from drying out, and they receive abundant food from the environment of the large intestine. Scientists have long noticed that bacteria, mitochondria, and chloroplasts are similar in size. We also know that bacteria have DNA and ribosomes, just like mitochondria and chloroplasts. Scientists believe that host cells and bacteria formed an endosymbiotic relationship when the host cells ingested both aerobic and autotrophic bacteria, cyanobacteria, but did not destroy them. Through many millions of years of evolution, these ingested bacteria became more specialized in their functions, with the aerobic bacteria becoming mitochondria and the autotrophic bacteria becoming chloroplasts. The central vacuole. Previously, we mentioned vacuoles as essential components of plant cells. If you look at figure 4.8b, you will see that plant cells each have a large central vacuole that occupies most of the cell's area. The central vacuole plays a key role in regulating the cell's concentration of water in changing environmental conditions. Have you ever noticed that if you forget to water a plant for a few days, it wilts? That's because as the water concentration in the soil becomes lower than the water concentration in the plant, water moves out of the central vacuoles and cytoplasm. As the central vacuole shrinks, it leaves the cell wall unsupported. This loss of support to the plant cell walls results in the wilted appearance. The central vacuole also supports the cell's expansion. When the central vacuole holds more water, the cell becomes larger without having to invest considerable energy in synthesizing new cytoplasm. 4.4. The endomembrane system and proteins. By the end of this section, you will be able to do the following. List the components of the endomembrane system. Recognize the relationship between the endomembrane system and its functions. The endomembrane system, endo equals within, is a group of membranes and organelles, figure 4.18, in eukaryotic cells that works together to modify, package, and transport lipids and proteins. It includes the nuclear envelope, lysosomes, and vesicles, which we have already mentioned, and the endoplasmic reticulum and Golgi apparatus, which we will cover shortly. Although not technically within the cell, the plasma membrane is included in the endomembrane system because, as you will see, it interacts with the other endomembranous organelles. The endomembrane system does not include either mitochondria or chloroplast membranes. 108. Chapter 4, Cell Structure. Access for free at openstax.org. Visual connection. Figure 4.18. Membrane and secretory proteins are synthesized in the rough endoplasmic reticulum, RER. The RER also sometimes modifies proteins. In this illustration, an attachment of a purple carbohydrate modifies a green integral membrane protein in the ER. Vesicles with the integral protein bud from the and fuse with the Golgi apparatus cis face. As the protein passes along the Golgi cisternae, the addition of more carbohydrates further modifies it. After its synthesis is complete, it exits as an integral membrane protein of the vesicle that buds from the Golgi's transface. When the vesicle fuses with the cell membrane, the protein becomes an integral portion of that cell membrane. Credit. Modification of work by Magnus Mangsky. If a peripheral membrane protein were synthesized in the lumen, inside, of the would it end up on the inside or outside of the plasma membrane? The endoplasmic reticulum. The endoplasmic reticulum, figure 4.18, is a series of interconnected membranous sacs and tubules that collectively modifies proteins and synthesizes lipids. However, these two functions take place in separate areas of the the rough and the smooth respectively. We call the tubule's hollow portion the lumen or cisternal space. The ER's membrane, which is a phospholipid bilayer embedded with proteins, is continuous with the nuclear envelope. Rough scientists have named the rough endoplasmic reticulum, RER, as such because the ribosomes attached to its cytoplasmic 4.4, the endomembrane system and protein's 109 surface give it a studded appearance when viewing it through an electron microscope, figure 4.19. Figure 4.19. This transmission electron micrograph shows the rough endoplasmic reticulum and other organelles in a pancreatic cell. Credit. Modification of work by Louisa Howard. Ribosomes transfer their newly synthesized proteins into the RER's lumen where they undergo structural modifications, such as folding or acquiring side chains. These modified proteins incorporate into cellular membranes, the or the ER's or other organelles' membranes. The proteins can also secrete from the cell, such as protein hormones, enzymes. The RER also makes phospholipids for cellular membranes. If the phospholipids or modified proteins are not destined to stay in the RER, they will reach their destinations via transport vesicles that bud from the RER's membrane, figure 4.18. Since the RER is engaged in modifying proteins, such as enzymes, for example, that secrete from the cell, you would be correct in assuming that the RER is abundant in cells that secrete proteins. This is the case with liver cells, for example. The smooth endoplasmic reticulum, SER, is continuous with the RER but has few or no ribosomes on its cytoplasmic surface, figure 4.18. SER functions include synthesis of carbohydrates, lipids, and steroid hormones, detoxification of medications and poisons, and storing calcium ions. In muscle cells, a specialized SER, the sarcoplasmic reticulum, is responsible for storing calcium ions that are needed to trigger the muscle cell's coordinated contractions. Link to learning. You can watch an excellent animation of the endomembrane system here. http://openstax.org/l/insane_in_the_endomembrane . At the end of the animation, there is a short self-assessment. Career connection. Cardiologist. Heart disease is the leading cause of death in the United States. This is primarily due to our sedentary lifestyle and our high trans fat diets. Heart failure is just one of many disabling heart conditions. Heart failure does not mean that the heart has stopped working. Rather, it means that the heart can't pump with sufficient force to transport oxygenated blood to all the vital organs. Left untreated, heart failure can lead to kidney failure and other organ failure. Cardiac muscle tissue comprises the heart's wall. Heart failure occurs when cardiac muscle cells' endoplasmic reticula do not function properly. As a result, an insufficient number of calcium ions are available to trigger a sufficient contractile force. Cardiologists, cardi equals heart. Ologist equals one who studies, are doctors who specialize in treating heart diseases, including heart failure. Cardiologists can diagnose heart failure via a physical examination, results from an electrocardiogram, ECG, a test that measures the heart's electrical activity, a chest X-ray to see whether the heart is enlarged, and other tests. If the cardiologist diagnoses heart failure, he or she will typically prescribe appropriate medications and recommend a reduced Table 110 Chapter 4, cell structure access for free at openstax.org. salt intake and a supervised exercise program. The Golgi apparatus. We have already mentioned that vesicles can bud from the and transport their contents elsewhere, but where do the vesicles go? Before reaching their final destination, the lipids or proteins within the transport vesicles still need sorting, packaging, and tagging so that they end up in the right place. Sorting, tagging, packaging, and distributing lipids and proteins takes place in the Golgi apparatus, also called the Golgi body, a series of flattened membranes, Figure 4.20. Figure 4.20. The Golgi apparatus in this white blood cell is visible as a stack of semicircular, flattened rings in the lower portion of the image. You can see several vesicles near the Golgi apparatus. Credit. Modification of work by Louisa Howard. We call the Golgi apparatus the cis face. The opposite side is the trans face. The transport vesicles that formed from the travel to the cis face, fuse with it, and empty their contents into the Golgi apparatus lumen. As the proteins and lipids travel through the Golgi, they undergo further modifications that allow them to be sorted. The most frequent modification is adding short sugar molecule chains. These newly modified proteins and lipids then tag with phosphate groups or other small molecules in order to travel to their proper destinations. Finally, the modified and tagged proteins are packaged into secretory vesicles that bud from the Golgi's trans face. While some of these vesicles deposit their contents into other cell parts where they will be used, other secretory vesicles fuse with the plasma membrane and release their contents outside the cell. In another example of form-following function, cells that engage in a great deal of secretory activity, such as salivary gland cells that secrete digestive enzymes or immune system cells that secrete antibodies, have an abundance of Golgi. In plant cells, the Golgi apparatus has the additional role of synthesizing polysaccharides, some of which are incorporated into the cell wall and some of which other cell parts use. Career connection geneticist. Many diseases arise from genetic mutations that prevent synthesizing critical proteins. One such disease is Lowe disease, or oculocerebrorenal syndrome, because it affects the eyes, brain, and kidneys. In Lowe disease, there is a deficiency in an enzyme localized to the Golgi apparatus. Children with Lowe disease are born with cataracts, typically develop kidney disease after the first year of life, and may have impaired mental abilities. A mutation on the X chromosome causes Lowe disease. The X chromosome is one of the two human sex chromosomes, as these chromosomes determine a person's sex. Females possess two X chromosomes while males possess one X and one Y chromosome. In females, the genes on only one of the two X chromosomes are expressed. Females who carry the Lowe disease gene on one of their X chromosomes are carriers and do not show symptoms of the disease. However, males only have one X chromosome and the genes on this chromosome are always expressed. Therefore, males will always have Lowe disease if their X chromosome carries the Lowe disease gene. Geneticists have identified the mutated gene's location, as well as many other mutation locations that cause genetic diseases. Through prenatal testing, a woman can find out if the fetus she is carrying may be afflicted with one of several genetic diseases. 4.4. The Endomembrane System and Proteins. 111 geneticists analyze prenatal genetic test results and may counsel pregnant women on available options. They may also conduct genetic research that leads to new drugs or foods, or perform DNA analyses for forensic investigations. Lysosomes. In addition to their role as the digestive component and organelle recycling facility of animal cells, lysosomes are part of the endomembrane system. Lysosomes also use their hydrolytic enzymes to destroy pathogens, disease-causing organisms, that might enter the cell. A good example of this occurs in macrophages, a group of white blood cells which are part of your body's immune system. In a process that scientists call phagocytosis or endocytosis, a section of the macrophage's plasma membrane invaginates, folds in, and engulfs a pathogen. The invaginated section, with the pathogen inside, then pinches itself off from the plasma membrane and becomes a vesicle. The vesicle fuses with a lysosome. The lysosome's hydrolytic enzymes then destroy the pathogen. Figure 4.21. Figure 4.21. A macrophage has engulfed, phagocytized, a potentially pathogenic bacterium and then fuses with lysosomes within the cell to destroy the pathogen. Other organelles are present in the cell, but for simplicity we do not show them. 4.5. The cytoskeleton. By the end of this section, You will be able to do the following. Describe the cytoskeleton, compare the roles of microfilaments, intermediate filaments, and microtubules. Compare and contrast cilia and flagella. Summarize the differences among the components of prokaryotic cells, animal cells, and plant cells. If you were to remove all the organelles from a cell, would the plasma membrane and the cytoplasm be the only components left? No, within the cytoplasm, there would still be ions and organic molecules, plus a network of protein fibers that help maintain the cell's shape, secure some organelles in specific positions, allow cytoplasm and vesicles to move within the cell, and enable cells within multicellular organisms to move. Collectively, scientists call this network of protein fibers the cytoskeleton. There are three types of fibers within the cytoskeleton: microfilaments, intermediate filaments, and microtubules, figure 4.22. Here, we will examine each. 112 Chapter 4, Cell Structure Access for free at OpenStax.org. Figure 4.22 Microfilaments thicken the cortex around the cell's inner edge. Like rubber bands, they resist tension. There are microtubules in the cell's interior where they maintain their shape by resisting compressive forces. There are intermediate filaments throughout the cell that hold organelles in place. Microfilaments of the three types of protein fibers in the cytoskeleton, microfilaments are the narrowest. They function in cellular movement, have a diameter of about 7 nanometers, and are comprised of two globular protein intertwined strands, which we call actin, figure 4.23. For this reason, we also call microfilaments actin filaments. 4.5. The cytoskeleton 113 figure 4.232 intertwined actin strands comprise microfilaments. ATP powers actin to assemble its filamentous form, which serves as a track for the movement of a motor protein we call myosin. This enables actin to engage in cellular events requiring motion, such as cell division in eukaryotic cells and cytoplasmic streaming, which is the cell cytoplasm's circular movement in plant cells. Actin and myosin are plentiful in muscle cells. When your actin and myosin filaments slide past each other, your muscles contract. Microfilaments also provide some rigidity and shape to the cell. They can depolymerize, disassemble, and reform quickly, thus enabling a cell to change its shape and move. White blood cells, your body's infection-fighting cells, make good use of this ability. They can move to an infection site and phagocytize the pathogen. Link to learning to see an example of a white blood cell in action. Watch a short time-lapse video of the cell capturing two bacteria. It engulfs one and then moves on to the other. Click to view content, https://www.openstax.org/l/chasing_bacteria intermediate filaments. Several strands of fibrous proteins that are wound together comprise intermediate filaments, figure 4.24. Cytoskeleton elements get their name from the fact that their diameter, 8 to 10 nanometers, is between those of microfilaments and microtubules. Figure 4.24 Intermediate filaments consist of several intertwined strands of fibrous proteins. Intermediate filaments have no role in cell movement. Their function is purely structural. They bear tension, thus maintaining the cell's shape, and anchor the nucleus and other organelles in place. Figure 4.22 shows how intermediate filaments create a supportive scaffolding inside the cell. 114 Chapter 4, Cell Structure Access for free at OpenStax.org. The intermediate filaments are the most diverse group of cytoskeletal elements. Several fibrous protein types are in the intermediate filaments. You are probably most familiar with keratin, the fibrous protein that strengthens your hair, nails, and the skin's epidermis. Microtubules, as their name implies, microtubules are small hollow tubes. Polymerized dimers of alpha-tubulin and beta-tubulin, two globular proteins, comprise the microtubule's walls, figure 4.25. With a diameter of about 25 nanometers, microtubules are cytoskeleton's widest components. They help the cell resist compression, provide a track along which vesicles move through the cell, and pull replicated chromosomes to opposite ends of a dividing cell. Like microfilaments, microtubules can disassemble and reform quickly. Figure 4.25 Microtubules are hollow. Their walls consist of 13 polymerized dimers of alpha-tubulin and beta-tubulin, right image. The left image shows the tube's molecular structure. Microtubules are also the structural elements of flagella, cilia, and centrioles. The The latter are the centrosome's two perpendicular bodies. In animal cells, the centrosome is the microtubule organizing center. In eukaryotic cells, flagella and cilia are quite different structurally from their counterparts in prokaryotes, as we discuss below. Flagella and cilia. The flagella, singular equals flagellum, are long, hair-like structures that extend from the plasma membrane and enable an entire cell to move. For example, sperm, euglena, and some prokaryotes. When present, the cell has just one flagellum or a few flagella. However, when cilia, singular equals cilium, are present, many of them extend along the plasma membrane's entire surface. They are short, hair-like structures that move entire cells, such as paramecia, or substances along the cell's outer surface. For example, the cilia of cells lining the fallopian tubes that move the ovum toward the uterus, or cilia lining the cells of the respiratory tract that trap particulate matter and move it toward your nostrils. Despite their differences in length and number, flagella and cilia share a common structural arrangement of microtubules called a 9+2 array. This is an appropriate name because a single flagellum or cilium is made of a ring of 9 microtubule doublets, surrounding a single microtubule doublet in the center, figure 4.26. 4.5. The cytoskeleton 115 figure 4.26. This transmission electron micrograph of two flagella shows the microtubules 9+2 array. 9 microtubule doublets surround a single microtubule doublet. Credit. Modification of work by Dartmouth Electron Microscope Facility, Dartmouth College. Scale bar data from Matt Russell. You have now completed a broad survey of prokaryotic and eukaryotic cell components. For a summary of cellular components in prokaryotic and eukaryotic cells, see Table 4.1.